Good morning, everyone, and welcome to my presentation. My name is Dr. Daniel Sabo, and I work at the Georgia Tech Research Institute on a variety of projects. Today, I'm going to be talking to you about my project, Immersion Chiller Media Characterization and Effects on Peracetic Acid Half-Life Rates. Now, I know when I say peracetic acid, or PAA, everyone probably thinks of something slightly different in regards to PAA. Some of you may think of how PAA is used throughout a processing facility, or some of you may think about purchasing PA, while others of you may even think of how it affects wastewater treatments. Just to make sure we're all on the same page today, however, I will be focusing on PAA that concerns the pre and main chiller. With that little primer, let's get started, shall we? There are two main goals of this research project. The first is how some of the most common chiller conditions, such as pH, PAA, and organic carbon, change over time within the pre and main chiller. Additionally, we are looking to understand how these conditions affect the stability and breakdown of PAA. The common chiller conditions that we focused on for this project include total suspended solids, total dissolved solids, fats, oils, and greases, chemical oxygen demand, and total kettle nitrogen. There have been both literature and anecdotal reports out there that state that PAA breaks down faster in the presence of some of these organic carbons. The extent of these effects are not well understood, which is what we are setting out to accomplish with this project. We're looking to understand not only which of the listed condition or conditions have the largest effect on PAA breakdown, but looking to see what component or components that make up those conditions is the culprit for rapid PAA decay. For the first part of this project, we visited two of our industrial partners and collected water samples from both the pre and main chiller every hour from before the first wog dropped until the last wog dropped, about 16 hours or so. Shown here is a simple diagram of the chilling process with a pre-chiller, main chiller, and finishing chiller. Marked in red letters of A, B, C, and D are the four locations that we collected water samples from. Two from the pre-chiller, where the wogs drop in and where they leave, and two in the main chiller, again, where they enter and leave the chiller. We performed some analysis on site, such as PAA and pH levels, and brought back samples to our lab for additional tests. We measured the total suspended solids, total dissolved solids, fats, oils, greases, chemical oxygen demand, and total kettle nitrogen back in our lab. For the PAA measurements, we used our own calorimetric absorption kit. Now, there have been questions about the comparability of this kit versus titration kits, which are commonly used in industry to measure the PAA levels throughout processing plants. Shown here are two charts that compare the two methods for the samples we collected from the pre and main chiller. As you can see, the two methods are pretty close for the main chiller and follow similar trends, which can be seen in the top graph. For the pre-chiller, there is more variability between the two methods. This is due to the fact that the titration kit, you are adding chemicals drop-wise until the sample color changes from a dark brown or blue color to clear. Depending on how the measurement is set up, one drop can represent up to 15 parts per million of PAA. This can cause large differences between the titration and calorimetric method. For reference, in the main chiller, one drop in the titration measurement was only 5 parts per million PAA, allowing for tighter measurements between the two kits. Now let's get to the data from the samples we collected. From the two plants that we collected samples from, we have an interesting set of data. One note on this data. I have shown the range of each individual organic carbon species, but have included an average of the species in parentheses if a steady state was reached. For example, in the fats, oils, and grease measurements, we started low and went high in the pre-chiller, reaching an equilibrium within a few hours, and would fluctuate around 140 to 200 parts per million for plants 2 and 1 respectively. On the other hand, the main chiller saw a continual, albeit slow, increase throughout the processing day for both plants. The chemical oxygen demand for both plants were similar in their trends and concentrations. In the pre-chiller in both plants, the concentration reached some steady state after 4 hours, but continually increased throughout the day in the main chiller, as expected. Similar trends were observed in the total suspended solids and total dissolved solids for both plants. In the pre-chiller, would reach an equilibrium within about 4 to 6 hours and would fluctuate around the average values seen in the parentheses. The concentrations for those same conditions would increase throughout the processing day in the main chiller. The total kettle nitrogen was an interesting component, and their charts could be seen below. For plant 1, similar trends that I just described can be seen in the bottom left image, where the pre-chiller reached an equilibrium within 4 hours, while the main chiller saw a continual increase throughout the processing day. In plant 2, both the pre and main chiller saw a continual increase throughout the day as seen in the picture on the bottom right. One last thing to note is that we're still working through plant 2's data, so the fats, oils, and grease number may change. But with that, it is interesting to know that most of these species are pretty similar to one another despite being from 2 different processing plants. We also performed an elemental analysis of the samples over time, as well as looked at the pH and PAA levels for both plants. For the elemental analysis, whose data can be seen in the top left-hand graph, we saw that the sodium, Na, phosphorus, P, and potassium, K, for both plants were very similar and showed similar trends throughout the day. They started low and continually increased, not reaching a maximum or plateauing by the end of the processing day. For the calcium Ca and magnesium Mg, we saw some stark differences. I zoomed in on those 2 elements for the bottom left-hand chart. As you can see that in plant 1, the calcium and magnesium levels are much lower than that of plant 2. This makes sense as one plant uses municipal water while the other uses well water, owing to increased hardness and higher calcium and magnesium levels. For the pH, both plants were similar in their levels of pH for both the pre and main chiller, and the data can be seen in the upper right-hand graph. In general, in the pre-chiller, there was an initial decrease for the first 3 to 4 hours, followed by a level off and slight fluctuations, as indicated by the small range bar in the middle of the bar graphs. For the main chiller, again there was an initial decrease for the first 3 or so hours. However, the fluctuations were greater compared to the pre-chiller, indicated by the larger range bars. And finally, for the PAA, we see some stark differences between the 2 plants, the data of which can be seen in the bottom right-hand graph. For plant 1, the average PAA levels were 30 to 40 parts per million for both the pre and main chiller. There was an initial rapid decrease in concentration for the first 3 hours, followed by a level off of concentration. From the range bars, it can be seen that the PAA concentrations would fluctuate widely throughout the day. For plant 2, the pre-chiller had an average of 150 parts per million, while the main chiller sat around 100 parts per million of PAA. Again, there was an initial decrease in PAA concentration for the first few hours. However, the drop wasn't as drastic as plant 1. Similarly to plant 1, the PAA concentrations would fluctuate widely throughout the day. For part 2 of our project, we wanted to see how the individual chiller media components would affect PAA stability and therefore breakdown. We used the information from part 1 of our project to direct the concentration of these individual components. We initially focused on the total suspended solids and total dissolved solids. We then started looking at the species that make up the TDS and their effect on PAA stability. The reason for this will become apparent shortly. For all of our tests, we adjusted the pH of our samples to maintain 9.0 throughout the experiments. We used an industrial-grade PAA from one of our partners. For all of our studies, we used our calorimetric absorption kit to measure all PAA concentration. Shown in the picture to the right is how the color that is measured changed throughout one of our tests. As you can see, the color lightens, indicating that the level of PAA is decreasing in each subsequent sample. For comparison for all of these measurements, I've calculated the half-life, or the time it takes for half the concentration of PAA to become consumed for potable water adjusted to 9.0. Under this condition, we found that the half-life was approximately 145 minutes. For the first species I looked at, the total suspended solids at 2,000 parts per million, we saw a half-life of approximately 80 minutes. Since the pH of these samples was maintained at 9.0, you can see that there is a small effect of TSS on the peracetic acid breakdown, as most of it is due to the pH adjustment. When we look at the total dissolved solids, we started with 3,500 parts per million and 1,750 parts per million. From this table, you can see that TDS has a huge effect on PAA stability. We have half-lives ranging from 2.5 to 6 minutes for the 2 TDS concentrations. With this interesting data, we started to investigate what makes up TDS and looking at their effects on peracetic acid. The first component of the total dissolved solids we looked at was proteins. So we took bovine serum albumin, also known as PSA protein powder, and dissolved it in water at levels similar to that found in part 1 of our project. We found that at the highest protein level, there was very little, if any, effect on PAA breakdown, with a half-life of approximately 141 minutes. Next, we looked at other components of TDS, focusing on nitrogen in the form of ammonia. From this study, we can see that nitrogen had a huge effect on pH stability with a half-life of only 1 minute. We are still in the middle of these experiments and have other species and contributing components to look at, but it looks like nitrogen in the TDS plays an important role in pH stability and breakdown. As I mentioned earlier in part 1 of this project, a majority of the carbon species in the pre-chiller will reach a steady state within a few hours, typically 4 to 6. On the other hand, those same species will continually increase in the main chiller throughout the processing day. As for the species that have the largest effect on pH stability, I showed that the TDS and by extension nitrogen cause the peracetic acid to rapidly decay. Next, we are planning to investigate the effects of fats, oils, greases, blood proteins, and cations on PAA decay. Finally, we're looking to develop a chart or table that shows the effects of these various organic carbon species on pH stability. With that, I would like to say thank you to my team who were instrumental to this project, to the Agricultural Technology Research Program here at GTRI for the opportunity and funding to perform these studies, and to all of you for your time and attention. I look forward to answering any questions you may have, so please feel free to contact me. My contact information can be found here. Thank you and have a wonderful day.